Further introduction, and he will talk with us about Seneca Valley virus. The floor is to you, Nick.
Good morning. Sorry to be talking to you about Seneca Valley virus, which is not foot and mouth disease virus. But as you will see from the presentation, it's going to be potentially an important virus, particularly for in differential diagnosis from foot and mouth disease virus. So the virus was discovered or isolated from 1988 through the early 2000s from a number of samples submitted from pigs to Ames in Iowa, the National Vet Service Laboratory, and also one or two submitted to Plum Island because they were apparently showing a vesicular disease. But all the early ones weren't associated with vesicular disease. They were just isolated incidentally when investigating other conditions. Some of these viruses were sent to me in 1999 and 2003. I did some partial sequencing on the polymerase and found that they were all the same virus. And at Ames, they had already made an antisera to one and shown that they were related. And then soon after that, a virus was isolated at a company called Neotropics in the States as a cell culture contaminant in some of their cells. And they sequenced the whole genome and sent that to me, and I compared it with mine and showed that it was the same virus. And they were using it as an anti-cancer agent, and it has been used in clinical trials, and that's all been published. So what we found is that Seneca Valley virus was a picornavirus with a fairly typical picornavirus genome structure, and this is part of the picornavirus tree and shows Seneca Valley virus where it fits. This is the P1 capsid tree, and here is foot and mouth disease virus and various other related viruses, Teshan disease virus, etc., and encephalomyocarditis, etc. So it fits in with kind of known picornaviruses, but is a separate genus called Seneca virus, and we've recently renamed the species Seneca virus A. But the common name is still Seneca Valley virus. It's a single serotype, and people started calling it Seneca virus A or Seneca A virus. That's not correct terminology in terms of the virus itself. So please remember that. The disease that's been shown in the field since the early observations, 1997 and early 2000s, of a possible associated vesicular disease, that has really advanced much more, and now we see it regularly causing vesicular disease, and you see the typical kind of lesions you get in pigs, which aren't necessarily as distinct as maybe foot and mouth diseases in cattle, etc. But these are, and there are plenty of papers showing these kind of lesions. Now recently, at Ames, it's been put back into pigs and shown experimentally to cause vesicular disease. And so we now have proof that this agent is the cause of a vesicular disease. And it's probably evolved to cause this vesicular disease from a condition where it wasn't showing it. And in fact, two earlier isolates were experimentally inoculated to pigs and didn't cause any disease, whereas this later one from 2015 is clearly showing vesicular disease. Now, the original virus isolates in 1988 were in Minnesota and North Carolina, and over those early years, then we had isolates coming from pigs in various places in the US, and with the explosion of more vesicular disease, there's probably many other states that are infected, but I haven't got that data. And this shows the density map for pig population in the US. And as we can see, the places where there are lots of pigs, there are also viruses being isolated, as well as in other other places. And then in 2007, pigs were moved from Minnesota into Manitoba and shown to cause a vescicular-like condition there. And more recently, they've been found in Ontario, 2016. And then in 2015, there were lots of reports of a vescicular disease in Brazil, various states in Brazil, and also in China in 2015, and more recently, 2016. Now, people have been sequencing the genomes of these viruses, and these are all the recent ones that have been sequenced by other people. But we, having had all these early viruses, we sequenced the complete genomes here. And what we have here is a maximum likelihood tree based on nearly the complete genome, because some people haven't necessarily gone to the ends. So we trimmed it back. So that's most of the genome. And as you can see, all the early isolates, and this is rooted on the earliest viruses. So what we can see is a progression of what is apparently a single lineage dating back into the 1980s. And most of these are from pigs, but there are a few exceptions. This one was the cell culture contaminant from neotropics, and then more recently on a farm in Minnesota, it was found in mice and in environmental samples, and they're all clearly related there. So it contaminates the environment quite easily. And this is one from Canada and China. Now, if we do some further analysis on this, and we have done Bayesian analysis, but I hadn't finished it to present it this time, but if we do analysis where we look at the date of isolation versus the genetic distance to the earliest isolates here, and plot those, we get an absolutely perfect straight line. It's probably one of the best I've ever seen for this type of evolution, showing that all these viruses are basically a single lineage. And you can date these back to around early 1985 as having a common ancestor. The neotropics one, because we didn't know when that had been introduced into cell culture, and it was found, sorry, where is it gone? There. It was found in about 2002. I dated that on the tree by placing it on the line and then reading it off, and it comes out in around 1999. So that's quite reasonable. Now, in the laboratory, the virus will grow in a number of cell lines, mainly pig kidney have been used, for example, IBRS2 or PK15. But it does grow in lamb kidney, grows in monkey cells and rabbit kidney cells. So the virus neutralization test can be used. I believe there's a competitive ELISA available in the US. And then there's been some conventional RT-PCRs targeting 3D, and then a real-time cyber-green-based assay targeting VP1. But there have been no real-time assays which target 3D, which is probably the most conserved part of the genome, or at least will be in the future as the virus evolves. So with the increase in the number of genomes available, not sure what to do with that, but okay. We've designed a real-time assay based on 3D and evaluated that. And here is the sensitivity. We used the published data that's on GenBank, and that includes partial sequences for some of ours. And there was only, in one of the primers, we had to make one ambiguity. And the other primer and the probe, they were absolutely conserved. And so what we have here is basically a cutoff CT value of about 36.8. And so this is quite a sensitive assay. And this was done on one of the recent isolates from Kansas. And then it's been tested on a number of Seneca Valley virus isolates collected between 1988 and 2015, and some various nasal and rectal swabs. And we found no false positives for the nasal swabs, some for the rectal swabs, but they were all above the diagnostic cutoff of 36. And the specificity was 100% using RNA extracted from foot and mouth SVD of vesicular stomatitis. And we also predicted, looking at vesicular exanthema, looking at the sequence, we predicted there wouldn't be any reaction, any cross-reaction with VESV either. So the summary of this bit of data is that we've got a new and rapid sensitive real-time RT-PCR, which could be used by reference laboratories to detect SVV. And because it's targeted on 3D, we expect it to be an assay which will last into the future, at least until the virus changes enough that it becomes a problem. And we can also use this for research, as a research tool for other studies. So we think that Seneca Valley virus is probably introduced into the US pigs in around 1985 from an unknown source. I mean, potentially that could be pigs in another country, but we have no evidence for that. Or it could be animals within the US, a different host species. For example, rodents on farms. But it appears to be a single introduction. So in US, SVV has occurred in most of the major pig-producing areas, spread to Canada at least in 2007, as far as we know, and it's recently spread to Brazil and China, and we are aware of one other country in Asia, which I can't say about at the moment, which also has it. Outbreaks of idiopathic vesicular disease were associated with SVV, but now with the infection in Ames, it's shown that this virus does cause vesicular disease. And in fact, interestingly, recently, 8 truckloads of pigs being exported from Ontario to the States were turned away at the border due to vesicular lesions. So diagnostic tests, assays, both molecular and virological, are now in place to allow the identification of this virus. And I think for the future, we would predict that SVD will spread around the world in the next few years. There is no real controls, and this may present problems with differential diagnosis with other vesicular diseases, principally foot and mouth disease virus. And I think searching for the origin of Seneca Valley virus would be certainly an interesting thing to do, and we would predict there will be more related viruses in whatever host species it originated in. And there's always the potential that it could spread again. And as we know, swine vesicular disease spread from humans into pigs on one occasion and caused a lot of problems. But actually, Coxsackie B4 also spread from humans into pigs in the 70s or before. Didn't cause so many problems, but did cause a fair bit of disease in Russia. So it is possible these things can happen when we least expect it. So I'd like to acknowledge Jim House and John Landgraf from Plum Island and Ames, who are both now retired, mainly for the virus isolates, and then the people at Kansas State University for their collaboration on the real-time assays, and Susanna Williamson at APHA, who is interested in having diagnostic capabilities in the UK for when the virus enters our country, and Valerie and her team for helping out with various viruses and assays and things. So thank you very much.
Thank you very much, Nick. Time for 1 or 2 questions. Yes, please, go ahead, Michelle.
Could you please emphasize the epidemiology of the virus? How does it spread from animal to animal, from farm to farm? Is it a fragile virus?
Well, we don't really know too much about the spread, but it looks like it contaminates the environment quite well. So and we would expect it to be transmitted pig to pig or by contamination of fomites, etc. It's said that it's not particularly prevalent, but again, I don't think the studies have really looked at that enough yet. And the disease, while obviously being obvious if it's true vesicular lesions, we don't know about how the virus may spread in a subclinical form. And there have been also associated mortality, which again, we're not sure whether is really true to that being caused by that virus. But I think that a lot of these questions have yet to be answered. Thanks.
Thank you. Nick, one short question. Did you test archival samples from the UK?
No, we haven't. We have tested a few samples that have come in from abroad, from Hong Kong, which were sent in for foot and mouth diagnosis, but were negative. And there were a few which had borderline CT values, so we're not quite sure whether they're positive or not. But the fact that it's now in China, we wouldn't be surprised to find it somewhere. But no, we haven't looked at the UK yet. And I think that we would have to do in conjunction with the APHA at some stage.
Okay, thank you.
All right, so thank you, Nick. Can I ask a question, please? Oh, make some comments. Sorry.
Who's talking?